And today we'll be investigating ecosystems, a part one of two lecture. So we're going to look at some types of ecosystems with some examples of each. So you can divide ecosystems into natural and artificial. Artificial simply meaning man-made. So under natural it may be terrestrial meaning on land or aquatic meaning related to a water body. Examples of terrestrial natural ecosystems: grasslands, forests or woodlands, and deserts. Examples of natural or aquatic ecosystems include oceans and lakes. And as we said, artificial or man-made ecosystems, we have aquarium or a crop field. So when we're studying ecosystems, we want to study the biotic, meaning the living elements, and the abiotic, meaning the non-living elements within the ecosystem. We want to look at the relationships between plants and animals and how they relate with abiotic factors. So we're going to look at an ecosystem in Jamaica as an example. So when you're looking at ecosystems or studying them, you want to have a scale outline map of the ecosystem that covers the area in which you'll be studying. So we have a little map right here that's highlighting the Blue Mountain National Park. So when studying ecosystems, we tend to want to determine the various species of organisms present and their respective numbers. It's almost impossible to single-handedly count all the members of all species. So we come up with methods of sampling to help us estimate population sizes. So the most inexpensive and straightforward sampling method is simply observation. So looking at the common species of plants and animals in their natural environment, looking how they adapt to their environment. For example, are they camouflaged? Do they have some symbiotic relationships occurring in the ecosystem? So another sampling method is QUADRATS. As the name suggests, 4 sides. It's usually a square frame of known area, either 0.25 meters squared or 1 meter squared. So when we're using quadrats, we tend to throw the quadrats at random areas within the ecosystem several times, and each time we count the number of each species that we find. Quadrats are used for plants and stationary or slow animals. Sometimes you may use a percentage of the quadrat area if it's impossible to distinguish the individual plants. Quadrats tend to be used in uniform ecosystems, for example, grasslands, which we said is an example of a natural terrestrial ecosystem. So, can quadrats be used to sample fauna? Well, yes. It's slow moving or sessile, and so it's easy for us to count it once we've thrown the quadrat into the area. But we do not use quadrats for fast-moving animals. As we said, if it's impossible to actually count the individual plant, we can use a percentage cover of the quadrat. And it's better done by one person to basically eliminate the biases. So, if we had a quadrat thrown and we found that almost half of the quadrat was covered, then we'd say that's about 50% coverage. So when we're looking at quadrats, we can use our results to determine the species density, which is the average number of individuals of each species per meter squared. We calculate this by looking at the total number of individuals of the species in all the quadrats thrown, and we divide that by the total number of quadrats thrown. Now it's important to note that when we're using quadrats, we want to count the species that actually fall within the quadrat. So in this case, the flower is about 80% is within the quadrat. So we're able to count this. But in this flower, about 90% is outside. So we would not count this. So species cover is a percentage of ground covered by a species if the individual organisms were difficult to count. We're going to look at this example with buttercups. So let's see about how many boxes are covered by the buttercups. These two together make about 1. See, we have a little space that this would fill. So this is about 1. This is also about 1. These two boxes together, 1. Same here. These two boxes make about 1 completely covered box. This would be 2 separate boxes. So far we've gone 1, 2, 3, 4, 5, and this is almost completely covered. So let's count this as 6. This is quite insignificant. So the buttercup is covering on average about 6 boxes out of the 25 boxes. So we'd say that the species cover is 6 out of 25, and we multiply that by 100 to get a percentage. So we would say that the buttercup is covering 24% of the area in which we were studying. So when we're looking at species frequency, which is the percentage of quadrats in which a particular species was found, we must first find the total number of quadrats containing the organism. Divide that by the total number of quadrats thrown, and to find it in percentage, we multiply by 100. Let's look at this example. So let's look at the frequency of bunch grass in this quadrat over here. So the bunch grass, as you can see depicted by the key, looks like this. So how many quadrats contain bunch grass? 1, 2, 3, 4. You notice I counted this as 1 because it's not about the number of organisms when you're looking at frequency. You just want to know how many quadrats contain at least one of the organism. So it's 1, 2, 3, 4. So the frequency of bunch grass over this side would be 4 over 36. And when we multiply that by 100, we get about 11%. Let's look at the cattail. How many quadrats contain the cattail? 1, 2, 3, 4, 5, 6, 7. So 7 quadrats. So 7 out of 36, that's about 19%. Let's look over here. The frequency of bunch grass in this case, 1, 2, 3. Only 3 quadrats contain at least one bunch grass out of the 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 total quadrats. So 3 out of the 9 contain bunch grass. The cattail, 1, 2, 3, 4, 5. So 5 out of the 9 contain cattail on this side. So the frequency is about 56%. So we're going to look at a worked example of species density. So we have the different quadrats that we threw, and we have the number of individuals found each time we threw the quadrat. So for example, in the first quadrat we had 5 individuals. In the third time we threw it, we had 4 individuals. And in the fifth time we threw it, we had 7 individuals. So when we're working out the density, it's the total number of individuals of species in all the quadrats divided by the number of quadrats and times that by the quadrat area. So the total number of individuals would be 5 plus 6 plus 4 plus 2 plus 7. And we would divide that by 5 because that's the number of quadrats thrown. And since we used a 1 meter squared quadrat, it's simply times by 1 meter squared. So we're going to look at species cover. So the percentage coverage is equal to the areal coverage of all the quadrats divided by the number of quadrats times by 100%. So in this case, the coverage is 9 plus 8 plus 8 plus 7 plus 8. And we divide that all by 5, multiplied by the quadrat area at the bottom, which is 1 meter squared. And we multiply everything by 100 to put it in percentage. So we're going to look at species frequency again. So we said that species frequency is just the percentage of sample quadrats that shows the species or the target group appearing. And we're using the slashes to suggest that the presence of the species and the 0 to represents the absence. So how many quadrats contain the species? Well, we have 1 here, 2, and 3. So 3 out of the 5 quadrats contain the species. So we have a bonus question. There are 4 dandelion plants inside a 0.25 meter squared quadrat. The whole field is 50 meters squared. What would the estimated population size of the dandelions in the field be? So we said there are 4 dandelion plants inside a 0.25 meter square quadrat. The whole field is 50 meters squared. What's the estimated population size of the dandelions in the field? So 800 dandelions. And how did we get this? So 4 dandelion plants were found in 0.25 meters squared. So in order to find the estimated amount in 50, we just simply multiply by 50 and we'll get 200 divided by 0.25 and we get about 800 dandelions would be expected in the 50 meter square field. So line transects are also used for stationary or slow animals. It's basically a horizontal line, usually made of string placed across the ecosystem or parts of the ecosystem. And we basically record the species of plants and stationary or slow animals touching the line at fixed intervals. For example, we may use a 15 centimeter interval each time. It's usually used when a habitat type changes into another, for example, a grassland merging into a woodland. Quadrats can be used with line transects. As you can see in the diagram, we have the quadrats at the various intervals. Line transects are used to study shrubs and tree layers of woodlands. So we can use the results from line transects to create a kite diagram which shows the number of animals or percentage cover of plants against the distance along a transect. So you can see on the x-axis we have the distance as you move along the line transect, and you can read on the y-axis the value that would show the percentage cover at each point along the distance. So the next sampling method is capture-recapture. As the name suggests, you're capturing the animal, releasing them, and recapturing them. It's used to estimate population sizes of animals which are moving. So you want to trap the organism you're studying, count it, and make a specific mark to represent it being in the first sample of animals. Some persons may mark with a spot of paint or a tag. You release the organism in the same area a day or more later. Then you're going to do the same process, trapping and then counting the second sample. Now when you're calculating the estimated population size, it will be the number of organisms in the first sample times the number of organisms in the second sample, and you divide that all by the number of marked organisms that were recaptured in the second sample. So we're going to look at an example of capture-recapture. So Blue Mountain National Reserve is trying to find out how many trout there are in the pond below the waterfall. They catch a sample of 5 trout and tag them. The researchers then release them back into the pond. The next day they catch another sample of 4 trout and find that one has already been tagged. What is the estimated population size of trout in the pond? So, we said already that the estimated population size is the number of organisms in the first sample, which from the question we knew were 5, times the number of organisms in the second sample, which we knew was 4. However, they said that one of the organisms was already marked. So, to calculate the estimated population size, it would be 5 times the 4 divided by the 1 trout that was recaptured. So the estimated population size is equal to 20 trout. 10 sharks were trapped, marked, and released. 2 days later, 20 sharks were trapped. 5 of these were found to be marked. What is the population size? So, once again, we started the question with 10 sharks being captured in the first sample, and we're going to multiply that by the 20 sharks captured in the second sample, and we want to divide that by the number of sharks that were already marked, meaning they would have belonged to the first sample, which the question said was 5. So it's 200 divided by 5. The estimated population size is 40 sharks. The next sampling method we're going to look at is sweep nets. These nets are simply used to sample flying insects and other small animals from bushes, long grass, and ponds. Basically, the researcher stands and sweeps the net at a fixed number of times at randomly selected places within the ecosystem. They're going to count the number of each species of insect. This sampling method can be used with quadrats or line transects. So we're going to look at ways that you can collect organisms for these experiments that study ecosystems. One way is pouters, which are small jars for collecting insects using suction. It has 2 tubes. One goes into your mouth so you can apply the suction, and the other goes over the insect so that it is sucked into the jar. A fine mesh is held over the end of the first tube, which stops you from swallowing the insect. Pitfall traps is often used to get a sample of small invertebrates living on the ground, such as beetles, spiders, and slugs. It consists of a container such as a yogurt carton, which is buried in the ground. The top of the container is level with the soil surface, and it is covered by a piece of wood with a slight gap to allow insects to climb in. It is important to check the trap regularly to avoid the animals escaping or being eaten before they are counted. So if you found this video interesting and liked it, don't forget to give a thumbs up, share with a classmate, and subscribe to the channel so you can get more videos.